Class 9 Science, Chapter The Fundamental Unit of Life, Module 1. Observe the following structures: tower, monuments, buildings. They look very different from each other. They differ in shape and size. But do they have any common properties? Yes, they all are made up of bricks. Bricks are the basic building blocks. Similarly, there are millions of living beings on this earth, living organisms. They also differ in size and shape, but they all are made up of same building blocks. These building blocks are called as cells. Building blocks of life. How do you define a cell? Cell is the common, basic, structural, and functional unit of living beings. Discovery of cells. In the year 1665, Robert Hooke discovered a thin slice of cork, that is the bark of a tree, under a self designed microscope. There are empty compartments which resembled the structure of honeycomb. He labeled them as cells. In Latin, cells means small rooms. Living organisms. Living organisms are of two types: unicellular, multicellular. Unicellular is made up of single cell. Multicellular is made of many cells. In unicellular organisms, all functions are carried out by the single cell. In multicellular, many cells group together in a single body and assume different functions in it to perform to form various body parts. Example of unicellular is Chlamydomonas, Paramecium, bacteria, etc. Example of multicellular are some fungi, plants, and animals. Cell theory. Different scientists propose different theories regarding the study of cells. It states that all plants and animals are composed of cells and cell is the basic unit of life. In 1885, R. Virchow modified this theory by suggesting that all cells arise from pre existing cells. With the discovery of electron microscope in 1940, it was easy to understand the complex structure of a cell. Module 2: Shapes of cells. Different cells have different shapes depending on their functions. Like Columnar, shaped like columns. Example, epithelial cells. Circular biconcave. Example, RBCs. Spindle shaped. Shaped like a spindle. Spindle is a device to spin fibers into threads. Example, smooth muscle cells. Oval. Example, ovum or egg cell. Irregular. Example, amoeba. Circular. Example, fat cells. Branched. Example, nerve cell. Spiral. Spring shaped. Example, spiral bacteria. Rectangular. Example, cells of Spirogyra. Rod shaped. Example, Bacillus bacteria. The shape and size of the cells are related to the specific function they perform. Cell structure. A cell is made up of different components like cell wall, found only in plants, plasma membrane, cytoplasm, cell organelles. Let us see them one by one. Plasma membrane. It is also called as cell membrane. It is the thin outer covering of a cell. It separates the contents of the cell from external environment. The plasma membrane is flexible and is made up of organic molecules called lipids and proteins. The plasma membrane allows or permits the entry and exit of some materials in and out of the cell. It also prevents the movement of some other materials. Therefore, the plasma membrane is called as a selectively permeable membrane. How does the movement of substances takes place inside and outside the cell? For example, when the concentration of CO2 inside the cell increases, the CO2 moves out of the cell by diffusion. So, diffusion means from higher concentration to lower concentration. Similarly, when the concentration of O2 inside the cell decreases, the O2 moves from outside to inside of the cell. Module 3. Plasma membrane also helps in the transport of water in and out of the cell. Let us see how exchange of water takes place in a cell through the plasma membrane. When the cell is placed in isotonic solution, means medium surrounding the cell has same water concentration as that of the cell. In this case, there is neither entry of water in the cell nor exit of water from the cell. So, no exchange of water in this case. So, the cell remains flaccid, means not firm nor hard. When the cell is placed in hypotonic solution, means medium surrounding the cell has higher water concentration than that of the cell, that is outside solution is very dilute. In this case, water moves inside the cell. So water enters inside the cell and it swells up. So the cell become turgid, means swollen. When the cell is placed in hypertonic solution, means medium surrounding the cell has lower water concentration than that of the cell, that is outside solution is very concentrated. In this case, water moves out of the cell. So water comes outside the cell and it shrinks up, that is plasmolysis. So the cell become plasmolyzed, that is shrinks. Hypotonic solution and hypertonic solution. So as you can see, in this case, water is entering inside the cell and cell is getting swollen. So here water moves from outside to inside. Here water moves from inside to outside. So we see that water always moves from a region of higher concentration to a region of lower concentration. Movement of water takes place through plasma membrane. This process is known as osmosis. So, how do you define osmosis? The passage of water from a region of high water concentration through a selectively permeable membrane, that is plasma membrane, to a region of low water concentration is called as osmosis. It is a pure mechanical process that doesn't require energy. Module 4. Osmosis is not the only method for exchange of materials in and out of the cell without spending energy. There are many more processes called as, the first one is diffusion. For example, taking in of oxygen from the air into the body and giving out carbon dioxide outside the body. Then how is osmosis different from diffusion? Diffusion and osmosis difference. Diffusion, it occurs in any medium. Osmosis, it requires liquid medium. Diffusion, the diffusing molecules can be solid, liquid, or gases. Osmosis, it involves movement of only solvent molecules. Diffusion, semi-permeable membrane is not required. Osmosis, semi-permeable membrane is required. The flexibility of cell membrane also enables the cells to engulf in food and other material from its external environment. The cell of amoeba engulfs food from the surrounding. This process is known as endocytosis, that is inside the cell. This process requires energy. The cell throws out waste and other secretions. This process is known as exocytosis outside the cell. This process requires energy. Module 6. Nucleus. Nucleus is a large spherical body generally lying at the center of the cell. It is most important part of the cell. It controls all the cellular activities and also plays an important role in cell division. It decides what function a cell will carry out after maturation. Nucleus is covered by a double layered covering called nuclear membrane. The nuclear membrane has pores which allow transfer of material from inside the nucleus to its outside, that is cytoplasm. The, nuc- the nucleoplasm contains one or more round shaped nucleoli, that is a singular nucleus. So, nucleoplasm means the jelly like fluid inside the nucleus. It also contains a network of fibers called chromatin fibers. Chromatin fibers are the thin thread like structures composed of DNA and protein. Chromosomes are visible as rod-shaped structures when the cell about to divide. Chromosomes are made up of DNA, that is deoxyribose nucleic acid. DNA modules transfer the hereditary information from one generation to the next. Hereditary means, for example, the color of the eye, color of the hair, etc. Functional segments of DNA are called genes. Generally, one gene controls one or more cell functions, but sometimes a single function is controlled by a set of genes. Functions of nucleus. It controls metabolic activities of the cell. If it is removed, the cell ultimately dies. Metabolic activities means the functions of the cell. Nucleus control cell division. It is involved in transmission of hereditary characters from parents to offsprings, means the children. Module 7. Endoplasmic reticulum, that is the ER. It is a large network of membrane bound, that is covered by the membrane, tubes, and sheets. Endoplasmic reticulum is of two types. One is rough endoplasmic reticulum, that is RER, and smooth endoplasmic reticulum, that is ACR. Rough endoplasmic reticulum, it looks rough under an electron microscope. It has particles called ribosomes attached to it. Ribosomes are site for protein synthesis. Smooth endoplasmic reticulum, that is ACR, it is without ribosomes. It manufactures fat molecules or lipids. Functions of endoplasmic reticulum. It helps in intracellular transport. It synthesizes proteins and lipids, that is fats, and transports them. Proteins and lipids synthesized by ear are used in building plasma membrane. This process is known as membrane biogenesis. Some lipids and proteins are used as enzymes and hormones. It transports proteins synthesized by ribosomes. ACR of certain cells of liver play a crucial role in detoxifying many poisons and drugs which enter our body through food, air, or water. Module 8. Golgi apparatus or Golgi complex. It was first described by a German scientist, Camillo Golgi. It comprises of membrane-bound vesicles approximately parallel to each other. Their sacs are called cistern and are filled with different enzymes. It is considered as the packing and forwarding department of the chemical factory of the cell. Functions of Golgi apparatus. The material synthesized near the endoplasmic reticulum is packaged and dispatched to various targets inside and outside the cell through the Golgi apparatus. Its function include the storage, modification, and packaging of products in vesicles. In some cases, it also converts simple sugars into complex sugar. The Golgi apparatus is also involved in the formation of lysosomes. Module 9. Lysosomes. They are simple, one membrane bound sacs filled with digestive enzymes. These enzymes are made by RER, that is rough endoplasmic reticulum. Due to various metabolic activities going on in the cell, organic waste is generated. The powerful enzymes are capable of digesting these wastes. So, lysosomes digest the waste. Plant cells have very few lysosomes because generally in most of the plants the waste is stored. Functions of lysosomes. As it is the digestive system of the cell, it protects the cell by destroying any foreign material like bacteria and viruses that invade the cell. They remove worn out cellular organelles and remove all organic debris. Worn out means the damaged, and organic debris means waste materials. This process is called autolysis, that is the self digestion. So they are called as demolition squads. During the disturbance in cellular metabolism, for example, when the cell gets old or damaged, lysosomes burst and the enzymes digest their own cells. Therefore, lysosomes are also called as suicide bags. Mitochondria. It is a double membrane structure. The outer membrane is porous, while the inner membrane is deeply folded. The folds are called cristae. They create a larger surface area for efficient functioning. The mitochondria have their own DNA and ribosomes. Therefore, they are capable of making some of their own protein. Mitochondria oxidize carbohydrates and fats with the help of enzyme to release energy in the form of ATP. Oxidize means breakdown. Functions of mitochondria. Mitochondria produce energy in the form of ATP, which is the energy currency of the cell. Energy currency means usable form of energy. Therefore, mitochondria are called as the powerhouse of the cell. The body uses this energy to synthesize chemical compounds. Synthesize means to produce. And to do mechanical work like movement of muscles, production of heat, conduction of nerve impulses, etc. Module 10. Plastids. Plastids are double membrane structures present only in plant cells. They give color to the leaves, flowers, and fruits. Plastids are of two types: colored and colorless or white. Colored plastids are called chromoplast, whereas colorless or white are called leucoplasts. So basically, plastids are of two types, that is chromoplast and leucoplast. Chloroplasts are a type of chromoplast which are green in color due to the presence of a green colored pigment called chlorophyll. Functions of plastids. Chloroplasts help to harness solar energy and convert it into chemical energy in the form of food. Harness means to trap. That means to trap the solar energy. Chromoplasts give color to flowers and fruits. Leucoplasts are involved in the synthesis and storage of various kinds of food like starch, oils, and proteins. Synthesis means production. Vacuoles. Vacuoles are storage sacs for solid or liquid contents. Vacuole is bound by a single membrane, that is, one layered membrane. They do not have any basic shape or size. The structure of the vacuole changes according to the need of the cell. Animal cells have few small sized and temporary vacuoles, while plant cells have one or more large vacuole. Functions of vacuoles. Vacuoles help to maintain osmotic pressure of the cell. Osmotic pressure means the balance of the water. They store metabolic byproducts and end products, that is glycogen, proteins and water. Byproducts means the products of various chemical reactions. They store waste products and food in animal cell, that is in amoeba it stores food, so it is called as food vacuole. In plant cells, vacuoles are full of cell sap and provide turgidity and rigidity to them. Sap means the mixture of mineral and water. Turgidity means the firmness, and the rigidity means the stiffness. Module 11. We have seen some important organelles of a cell. All the organelles were covered by a membrane. Cells having such membrane bound organelles are called eukaryotic cells, whereas the cells which do not have membrane bound organelles are called prokaryotic cells. Characteristics of eukaryotic cells. They have a well defined nucleus, that is with nuclear membrane, nucleolus and nucleoplasm. Size of the eukaryotic cell ranges from 5 to 100 micrometer, that is they are comparatively larger in size. They have more than one chromosome. They are present in higher advanced unicellular and multicellular plants and animals. Characteristic of prokaryotic cells. Prokaryotic cells have three basic structures, that is plasma membrane, cytoplasm and nuclear material, that is the DNA. Their nucleus lacks nuclear membrane, therefore their genetic material, that is a DNA, lies in direct contact with the cytoplasm. They contain a single chromosome. Such undefined region of the cytoplasm containing DNA is called nucleoid. Membrane bound organelles are absent. In photosynthetic bacteria or blue-green algae, plastids are absent. So their chlorophyll is present in vesicles. Photosynthetic bacteria means the bacteria which perform photosynthesis. Vesicles means the bag-like structures. Prokaryotic cells are smaller in size, ranging from 1 to 10 micrometer. Thank you. Please subscribe my channel to get more updated content.